Welcome, everyone. I'm Harvey Lodish. I've been on the MIT faculty for many, many years, and it's my great personal and professional pleasure to introduce Robert Dottin to you. Robert was a postdoc in my lab 40 years ago, which is a long time. I mean, it was before I think all of you were born. MIT biology then was building 16 in 56. The old cancer center had just opened. This building did not exist. Whitehead did not exist, wasn't even thought of. The Koch Cancer Center didn't exist. And a lot of the research in my lab was on Dictyostelium, which Robert will talk about because that's what he did in my lab and also for a good part of his professional career. He was born in Trinidad, which is an independent nation in the Caribbean, if you didn't know that. And he'll describe quite a bit of his early childhood. He was educated at St. Mary's Colleges in Trinidad, is that right?
It's a high school.
Oh, it's a high school. But then he received a scholarship to the University of Toronto, where he did his PhD, and then received a Centennial Fellowship, which is given, I think, only to the top 5 students in Canada each year to come to my lab. So I was delighted to have him. He'll describe the work he did on very early characterization of messenger RNAs in Dictyostelium, a typical eukaryote, single-celled eukaryote. He then went to Johns Hopkins University, where he served on the faculty for several years, and then was recruited to Hunter College, which certainly in the biological sciences and really in a large number of other areas is the premier undergraduate college of the City University of New York. And he's done outstanding research on Dictyostelium, which he'll talk about, but also set up a genomics computational biology center at Hunter, which is really the centerpiece for now a lot of work going on in the larger area that includes Rockefeller and Cornell medical schools, and also was very involved in recruiting minorities to Hunter and mentoring them, and then a program I'm particularly interested in learning about, which is Africa. So let me not take any more of my time and leave it to Robert, and basically start with your childhood. What was it like growing up in Trinidad? What was your family like? What was the country like? And how did you wind up in Toronto?
Well, thank you, Harvey. I feel privileged to be back here at MIT. 40 years seems just like yesterday. And I did postdoc work here, as you know. And I considered Harvey a brilliant scientist, quick thinking, and very confident. He still is. And inspiring as well. And a good leader. And so I benefited a lot from being here. And I'm grateful for that opportunity, which really helped my career a lot, as it will yours, when you do your postdocs and leave for other pastures. And thanks to Mandana and Linda for making this arrangement, and I'm very grateful to be here. I'll talk about my personal journey. I hope it will be sort of interesting and interactive, and please ask questions if you like, and we'll just go as quickly as we can. I'll talk about my early years in high school and the University of Toronto, and then MIT. Then I went to Johns Hopkins, and later on I moved to Hunter College in the City University of New York, which is where I still am, and some of the successes of the Gene Center that I helped to develop. And the Clinical Translational Research Center, which is a collaboration in which Hunter is a very big part with Memorial Sloan Kettering Cancer Center and the Weill Cornell Medical College and the Hospital for Special Surgery. And then, if we get time, I'll get to the Africa section. So I came from Trinidad and Tobago. It's a country with about 1.3 million people. And the distribution in the country is really odd. There are about 40% Afro-Caribbean people who live there. It's an independent nation now. And about 40% Indo-Caribbean, who came as indentured laborers after slavery was abolished. The Africans came as slaves. And indentured laborers just meant that they did the same thing as slaves, but they were granted land after a period of service. And so they make up about 80% of the population, and then you have people who are mixed. They call them Douglas. And then there are Europeans and Chinese, quite a significant population there. And my parents, my mother was a homemaker. She took care of the 4 kids. My dad worked as a merchant marine ship's engineer, and he was away for quite a bit of time. We saw him infrequently, but he had a great influence on our development. And my first fascination with science that I can remember was when my mom provided what I call a scientific explanation to assuage my primitive fears. I would wake up at night screaming at seeing shadows on the window moving around. And finally, my mother got very sick of that, and she took me in my pajamas out to the street, showed me there was a street lamp out there, there were trees down here, and then there was a window pane. And she indicated that light moved in straight lines, and that caused the shadows that I saw, and that was her scientific explanation. And she pointed out all of these features. She was not aware of Schrödinger's wave theory, and assumed that it was just a straight line that it moved, but it was sufficient to provide a very rational explanation to me, which inspired me in a way, and I still remember that event many, many years later. She's certainly not with us anymore. I cherish that rational scientific thinking. My oldest sister, I have 2 sisters, the older one, who is the brightest of us all, I think, she never went to college, and she has managed the family in a way because we are all over the world in a way, and she keeps us in contact with each other, and she is very aware of all the family issues and so on. I have an older brother who attended the University of the West Indies, and then he went to the University of Illinois. He does personnel management, and he has a master's degree from that University of Illinois. My younger sister is an accountant. She was trained in England and stayed there for many years. One time she heard the long-term forecast on the radio, and it said that there would be 50 years of bad weather, and then the weather would improve. And that's when she decided to leave England and went to live in Canada, where she still lives now and works, and she's an accountant there. She lives in Oakville, which is near to Toronto. And my own family is very small. My daughter, technically my stepdaughter, she's now 27 years old, and we just returned from a trip to Oxford and Paris, where she had visited as a child when I was on sabbaticals. And we also went to Barcelona, which was unfamiliar to her. But she could renew this kind of experiences, and of course we didn't go to Euro Disney at this time, but we did before. And so it was a very, I don't take vacations very often, and this was one that was very special. And we enjoyed it immensely. My son couldn't come because he was busy finalizing his job hunt and so on. And now he's 23 years old, graduated from the university, American University in Washington, D.C. And I raised him as a single parent after I got married and then divorced and remarried and so on. And when I lived in New York, he remained with me, and he was 3 years old at that time, and we've been together since then, for good and bad. He works as a junior programmer now in a company that does non-invasive diagnostics from DNA in maternal blood, which of course is a recent phenomenon that you probably know about from work that's been done around here too. And he's not doing the biological side, but he's doing some of the programming for that project. So I say I'm a single parent, and and I could say that I raised my son by myself, but that's of course in quotes. In reality, it took a whole village in a place called Irvington, New York, which is on the river, the Hudson River.
Rip Van Winkle.
Yes, very short. One village south of that, which is Tarrytown, and yeah. So, and that's where Washington Irving lived. That's true. With some help I got from my niece and my cousin and a whole village to raise this child. So naturally, I had to be both a soccer mom, going to all kinds of events, and to raise, and a professional scientist. And we faced a lot of challenges, let's say, multitasking, as some of you might find in your life later on with kids and so on. But together we went to international meetings and we shared the sabbatical in Oxford. He went to school there, left his school in Irvington, and remained connected through the internet. And then he had to, and then we returned back to his elementary school. He was about 8 or 9 years old. And he complained, of course, that he had 2 sets of homework to do, both in England and with his class, which he maintained all these contacts with. I was raised in a modest home in Trinidad, middle class, and by Trinidad standards, it's probably what you would call low income by American standards. And my parents had a strong emphasis on education and music. And I had limited skills in music, although my brother and sister played the piano, the violin and the piano respectively. So I turned the pages for the duets that they played for my father and things like that. I wanted to stop playing the piano that I made, and so much that I made a Faustian deal, which was that if I could stay at the top of my class, they would allow me not to play the piano, to drop my piano lessons. And unfortunately, my mother agreed to that, and I stayed at the top of the class. But now I regret that I'm not a jazz pianist anymore. So that's life. In high school, I was very keen on science, and I took there in Trinidad, which is sort of a British in its educational system. You can, instead of graduating in what would be your grade 12, you can do 2 extra years in what's called was then called A levels, advanced levels. And so I specialized in physics, chemistry, and zoology. And I taught myself calculus in order to learn physics, because I wasn't doing math, which is another advanced math, which was other students were taking. I formed a lot of strong friendships then, which I maintained even today. I'm in touch with some of my classmates who are now, some of them are retired businessmen now, doctors and lawyers, of course. And in fact, today I just got some texts from one of them who was in Toronto this time. So I think it's important to develop networks and to maintain them with even among yourselves here now, so that when you go out and you're into science and you start doing successes or you need advice and so on, you have these connections that can be very useful. That was one thing that I found lacking among minority scientists, because they're so far apart and they're unconnected and so on. And I'll talk a little bit about one solution that I developed for that. The schools in Trinidad were not segregated, and at that time, the Chinese and the whites and Indians and Afro-Trinidadians were equally brilliant, industrious, mediocre, or lazy, and as one another. And so we didn't have to worry about Asians beating the curve as people do in this in here. And we faced no stereotype threat, which was a kind of pressure that women and minorities experience in order to try to compensate and to outperform. And it's a way of sort of choking. There's a lot of research that's done by Claude Steele on this, and I would recommend that it's a fascinating. His book, which is called Whistling Vivaldi, is an excellent piece of work on that topic. So I received a scholarship from the government to go to a university anywhere in the world. And I chose to go to Canada because I had bad feelings about going to Britain. I knew lots of British people in Trinidad, and I really didn't like their attitude. It was very condescending. And I felt that if I went there at that time, it wouldn't serve me well. I didn't want to come to the United States because at that time it was just in turmoil with a lot of race riots and wars and all these things. Not much different from now, actually. And so I decided to choose the best university in Canada. At that time it was ranked 12th in the world, and I went to the University of Toronto. Many people think that McGill is the best university, but it's not. Well, it's not that. That was not the ranking. So I ended up in Toronto, and I chose to go to the University of Toronto, and I worked there. I was majoring in microbiology, and in summertime I had to find jobs and so on. I worked as a prospector for a large exploration company which was looking for copper or wherever they could find it, or any kind of mineral that was in large bodies. And so I spent the summer working in way 400 miles up into northwest Quebec, exploring as a sort of a junior geologist with other people on these lakes, a beautiful country, but just riddled with black flies that would eat you through your clothes. But it was a very enlightening experience for me. I spent another summer working as a longshoreman, which they call a stevedore, working, loading up trucks with refrigerators and things like that. But one summer I worked with Clarence First, who was my mentor for that program, a summer program. And many of you have probably had those kinds of opportunities, which are quite transformative. And so I did that, and we worked on regulation of gene expression in bacteriophage Lambda. And he was an excellent mentor, really very inspiring and very meticulous. I could never be as careful as he was, and was extremely patient. I could never be as patient as he was with me. And so those were things that were very valuable to me at that time. And I majored in microbiology honors and graduated with first-class honors. I was first in the class of science, and I also was first in the university. And that's how I got this centennial, or the Governor General's Medal, which was awarded to the person who topped. It was like summa cum laude, I guess, if that's what you call it here. Yeah.
Quote your thing, it's the best degree in the university.
So,
Don't be modest.
Well, I didn't say these things to boast about myself, which I'm not reluctant to do. But in fact, the point here, however, is that in spite of that fact, later on, I just wanted to emphasize the fact that I was well qualified for the things that I was doing. And I think many of you who are women, minorities, and so on, might find that this obstacle, that there's a perception even in spite of that, depending on the groups that you interact with, that you just came on a quota or something else like that. And so that's again something that induces stereotype threat in people, because you're trying to compensate all the time to show that you're really different and that you're not really one of those. And in fact, there's no reason why you shouldn't be on a quota in any case, if there's some disparity that's preventing you from developing as a scientist. So I got married to my first wife, and I continued studies in medical biophysics for my master's at the Ontario Cancer Institute in Toronto. It's also called the Princess Margaret Hospital. And there I worked with Clarence First again, and Lou Siminovich, both of whom had worked with Jacob and Monod. You probably heard of them, Nobel Prize winners in Paris, who performed a lot of breakthroughs on operons. I then did my PhD in medical genetics at the University of Toronto with Mark Pearson, who was my advisor, and I finished that in 1974. We worked with crude extracts and purified proteins, which took a long time. We didn't have restriction enzymes and all these fancy ways, and no kits. We couldn't buy all the reagents from even the tris buffer that people buy now from these companies at an exorbitant price. So that's how we did science at that time, in the Stone Age. And I worked on a system that had been developed by Jeffrey Zube at Columbia. And this was to allow DNA-dependent protein synthesis. So you'd take your DNA from Lambda, add large amounts of it to your extract, and in the presence of radioactive amino acid, you would see which proteins were made. And you could display these on gels. And so this, Zube was using had done this to look at some of the beta-galactosidase synthesis and so on. So we use this to assay regulatory proteins. And we found that when we looked at the proteins that were made of the template Lambda, we saw lots of proteins were made, and if you added some regulators, like the N protein, which is a positive regulator, you could see other proteins. It's an indication of what genes were being regulated by, the N protein. And that was a fairly sophisticated approach at that time in using cell extract in vitro. And that's how we really studied regulation in vitro. So then we wanted to look at the effect of the Lambda repressor, which, as you know, is a protein that turns off genes. And we got purified protein, Lambda repressor protein from Dale Kaiser at Stanford, added this to our extracts, and could show that, looking at all the proteins that are seen on the gel, that several proteins were turned off as a result. It's, in other words, the system that we were using was working properly, and the Lambda repressor was turning off the synthesis of lots of proteins from the DNA template. Lo and behold, we saw that another protein was actually being activated and turned on and synthesized. The more Lambda repressor we added, the more of this we saw in its radioactive form. And this was a very important discovery for us. It shows that the Lambda repressor induces, it's not just a repressor, but a positive regulator of its own synthesis. We could identify that the new protein that was made as the Lambda repressor by using mutants, templates, and so on. And that meant that a protein which is a negative regulator for a whole set of cascades in gene expression off of this simple system was, in order to maintain its functioning, had to turn itself on. And so this was a very important example of what occurs now with stem cells and other regulatory systems. You have to have a mechanism that's circular in a way, so that certain proteins, like this repressor, must turn itself on, or it must induce a synthesis of other proteins that will turn it on, turn it on, sorry. And in that way, regulatory proteins can maintain their functioning. And this is how stem cells, as you know from the work of Yamagana, there are maybe 4 transcription factors which were required in order to maintain stem cells from undifferentiated cells. So that was a significant finding. We did write up a paper about this and submitted it for publication before I left. And I spoke about it at many conferences and so on. And I was going to come to MIT, and of course I had been in contact with Mark Potashny, who had been working on the Lambda repressor, and we gave him eventually the manuscript with our work, but of course we made sure that it was published or accepted for publication. And it's good to do that. His work later showed that the repressor actually stimulates its transcription, and he developed all of these models of how the switch works and so on, and that's a really very exciting work that is important even today, because again it shows how regulatory proteins like this must have some way of persisting, and he of course argues that that other kinds of modifications which people call epigenetic, and he doesn't like the broad use of that term, are not modifications that are sustained, or in some cases are even found in flies or other organisms, so that the DNA methylation may not be. And he's written an article, which I'm sure some of you have read, about this dilemma. And histone modification may not be specific enough. These regulators are very specific. The Lambda repressor is very specific. It turns itself on. It's also specific because it shuts off the expression of a specific set of genes. And methylation is not specific. And so I would say that there's a lot more work that needs to be done in order to find out how that specificity is maintained in epigenetics. So I'll show you a few slides. I sort of jumped ahead. So that's my son, who's about 1 year old, observing the computer, and my daughter, she's about 4 years old at that time, and that's the family. And later on, we sort of move forward in a time machine, and you'll see there they are. He's just graduated from American University, and my daughter is she's now a nurse and still going on to do more degrees. That was a significant finding, which we published long ago, and with Mark Pearson, who was my advisor, and Lynn Cutler, who was a technician in the lab. And so that was called at the time autogenous regulation, and it's auto-regulation, and that's what many people still call it. Robert's article in 2011 talks about how it maintains memory by transcriptional auto-regulation. So science is competitive, and you need to make sure that you publish in widely read journals, and that your work is recognized, which means you need to talk about it, and that helps you to get other positions later on. You don't want the impression to be left that somebody who's done, that other people get credit for your work. And I'm not trying to overemphasize that point. As you know, George Lemaitre, who was the astronomer and an MIT graduate, right, had discovered that the universe was expanding. And he calculated the rate at which it expands in megapars per second and so on. He was a priest as well. He attended MIT for a while in 1927, I believe. And many, even today, although Lemaitre was the major person who did this discovery, sometimes the literature gets confused, and those discoveries are often attributed to Hubble. And I'm not sure why that happened, but it took many years before that was ever corrected. And he had a collaborator whose name was it Friedman? I believe Friedman from Russia. Lemaitre was from Belgium, I believe. And I've never found out how he did at MIT and why he chose to come here. But it's something I'd like to explore. I have a big interest in the cosmos and so on. So anyway, I went to, I had to decide where I wanted to go. I was attracted by gene regulation, and I read papers by Harvey Lodish. He'd published about Dictyostelium being a very important system for looking at gene expression in eukaryotes because it was a simple differentiating system. And it was very attractive to me. I won a fellowship which is called a Centennial Fellowship in Canada. And at that time they gave 5 of them to MDs and PhDs. And I got one of those, and I decided I wanted to come to MIT. I wrote to Harvey, and he accepted me. And so that's how I ended up here. I lived in Cambridge Port, which it's somewhere around here, I guess. Or maybe it's overtaken by all these buildings now, new buildings. On Pearl Street, anybody knows where that is? Oh, so it's still around. I remember being sometimes going to Titi the Bear, which is a bar around there, where Bonnie Raitt used to play music. I don't know if these things mean anything to you. It does? Surprised. Anyway, and I enjoyed my time here. I found Harvey, as I indicated before, available, very stimulating, and very knowledgeable. And so that those are things which good mentors provide. And I enjoyed my interactions. I met John King. Is he still here? He's still here, right? Jonathan King. I was inspired by him. He had a very sort of social view about science. And he and John Beckwith, who was at Harvard at the time. And they encouraged a lot of conversation about what science is going to do and what its social impact might be and so on. And it triggered a lot of thinking on my part. I felt privileged to be at MIT. I was working with some of the best people, and I met a lot of very good students. Lydia Villakamarov was here at that time, and Alan Weiner and Jack Rose and John Bergman, Pamela Langer was a student. Alan, Jack, and John were, John was a student as well with you. But Alan and Jack were postdoctoral fellows. So I was attracted by the promise of Dictyostelium. It was so simple, and it seemed that it was going to be a very unique developing system because the cells aggregate and form a multicellular organism. And so I think it's important to pick the right problem to do. I wouldn't pick Dictyostelium if I were doing it now, but at that time it was very important, I think. And the reason why I wouldn't pick it now is because the genome is not as stable as it seemed at the time, and many different strains that people were working with had different chromosomes and things like that. It makes it very confusing. And also we found that when we try to do RNA interference, it's very sort of touch and go. But those are things that happen much later, and of course you don't know at the time which system is the best one to work on. And I think that most people here would probably be working on human cells and things like that, which I didn't really see as being amenable to the kinds of manipulation that you could do then. Now I think many of them can be done. But that's not to encourage you all to go and work on human cells. I think that there's a lot to be done, as you know, in things like metagenomics and all of the fields that are so wanting for research activities. They were heady times, as I call them. David Baltimore was there at the time in Building 16, and we had lots of results coming out of these labs and so on. I did some research on caps, which are found at the 5' end, and that seemed to be really exciting at the time. of messenger RNAs, and discovered a different kind of cap that was found in Dictyostelium, and which was the only kind that could be found, not the other more complex form. And we hoped that this would have some regulatory significance. So we published a paper in Cell, and that paper was quite. I benefited a lot from that.
It was an important paper.
It was at the time.
At the time, but that's the whole point.
Exactly right. And so we did that work. It was, we had to work quickly. And this is one of the things that sort of frustrates. It's a yin and yang kind of thing for me in science, because I always felt that if I didn't do this particular experiment on the Lambda repressor or something like that, or on caps or whatever, that within a year somebody else would have done it, because science is moving so rapidly and there's so many smart people there doing it. So in a way, it felt, that, well, when are you going to do something that no one else can do? And I always felt that kind of rub about doing science. But of course, it's very satisfying and seductive to do the experiments that anyway, even if you know that other people are going to be doing them. And so that, for me, I don't know if other people feel that way about doing experiments. It's not a cynical view, but it really pushes you to be very efficient and to work very fast and so on. And, but it also means that, are you that special? And, if you're sort of egotistical a little bit, which I think we all are, then you might, you always want to do something, to have an idea that's really different from everybody else. And so, but those things come very infrequently, don't they? So anyway, we did the work on caps and so on, and we published that, and it was significant at the time. At MIT, I was also inspired by a technician whose name I just cannot remember, encouraged me to. There was turmoil going on in Boston. The schools were lousy, high schools, and there were a lot of resistance to busing was taking place. And the person who was adjudicating this whole thing was a judge whose name was Garrity, and you might have heard of him. And he's died recently, I think. And he lived in Belmont, although he was in charge of this district, and therefore his responsibility was to decide whether this was a constitutional problem and that there was any violation of the Constitution that was preventing black and brown people from getting access to quality education, a very familiar problem even today. And so he was trying to desegregate the schools in Boston, and a technician asked me about that and asked me whether I would be interested in helping in that effort. And because the judge needed to get some feedback about what was going on in these schools and so on. So I spent a little time, not that much, and I don't know if Harvey ever knew that.
I did not know that. I would have encouraged you, I suppose. But, I mean,
yeah.
Busing was a huge, very, very damaging process to Boston. Well, you can describe it, but...
Yeah. So I went with a police escort into some of the schools, and in South Boston, which is a dangerous area for people like me, and we went into the schools and sort of looked around to see what the physical plant was like and the atmosphere and all those kinds of things, and to try to provide more factual information. He was trying to create magnet schools in which people would leave their rundown schools and go to these magnet schools which offered more, and for black and white people. But there was a lot of resistance to that in South Boston. And
That's putting it mildly.
Yeah. And it was just awful. And it continued for many years. My role was very limited. I went a few times, and I tried to I didn't try to intervene. I just had to kind of write reports about what was going on and so on. People, well, they knew that I was there and why I was there, and so nobody really attempted to interfere because, of course, there were policemen around and so on. And not that that's really a source of comfort. So. But anyway, so I made my reports and that kind of thing. That was my contribution. I was really, that was a very important part of my political consciousness and my conscience after that. And that was a sort of a moment that really changed my thinking about education, because I had had all this elitist education myself, and never really understood until then, how privileged I was. And these are the people who were just going to these lousy schools and sort of pitted against each other would never make it as scientists, because they never had the kind of resources, exposure, encouragement, and all the other kinds of things that I had had. And so that was really a moment that changed me. And that bitter struggle ensued for many years. I think there were 100,000 people, of which in that district, of which about more than 50% were white. And by 1988, it had gone down to like 15% white. People put their kids in private schools and so on, and the schools, and nothing really changed. And I still don't know what the solution to these problems might be. But it was a first-hand experience that I had, and it was very moving. Just toward the end of my time here, I went to Copenhagen because I had been invited to come there a few years before that, and I went to work in Ole Moller's lab with Niels Fiel and other scientists there. I really enjoyed it. Bill Hazeltine had been there just before me, and they had a program in which scientists would come from America and work for a short time. That was a very enlightening experience. I wasn't in the sort of pressure cooker environment, and I enjoyed that immensely. I didn't speak Danish, but then I discovered that English is really the universal language for scientists. And wherever I lived in Europe, in Prague, Copenhagen, Paris, Germany, whatever, people many times they didn't even want to you're working, collaborating with people, and they want to learn English too, because that's how they, and so you would struggle to kind of talk in their language, but they wanted to hear you speaking in English. So it was a kind of an interesting dilemma. I enjoyed that interaction, and then I got back to the States and decided, and had to look for a job and so on. Did seminars around the country, went to and eventually decided I would go to Johns Hopkins University. Phil Hartman and Saul Roseman were prominent people in that department, in the biology department, and they welcomed me. And
this was Hopkins' Homewood campus,
Homewood
not
campus,
the medical school.
Right, yeah. The medical school was in East Baltimore. The Homewood campus was in a sort of a pristine part of the city. And like many medical schools, they tended to be in rather impoverished areas. And again, I was sort of in a select environment where people were very proud of, their place in life and that kind of thing. And oh, sorry, I didn't remember. Turn this up. And Peter Devriotes worked in that area, in the medical school. He worked on Dictyostelium as well. And he had been characterizing receptors of Dictyostelium and could show that cyclic AMP, which was an external agonist, could bind to cell surface receptors and would trigger events in the cell, which might result in chemotaxis. The cells all move together to form this multicellular organism from a single cellular organism. And this was very elegant work that he had been doing. And of course, there were other labs like Gilman and so on who were looking at the activation by receptors of the synthesis of second messengers in the cell. So that's the state of signal transduction at the time. I wondered, we were working using marker genes to look at development and so on, and we knew that these genes were turned on during development. And what I decided I wanted to look at was to find out whether in fact those genes were turned on by the receptor directly. And so we used a lot of analogs of cyclic AMP, which was the agonist, and used the ones that were specific only to the cell surface receptor and not to any intracellular proteins. And tried to show that the receptor itself had to be activated in order for genes to be induced. There were no examples, perhaps one example then, of receptor activation turning on genes, and that was from some oncogene. And so we conducted and designed a whole set of experiments with Badaluri, Harry Babu, and myself, who was a postdoc at the time. And we showed that we could activate gene expression by triggering the receptors on the cell membrane. So that was an early paper in signal transduction, which I thought was significant. I think I put that there. Because it showed that a pathway in which gene induction would take place. Later on, it was difficult to publish that paper because people were so skeptical that was occurring. And in fact, one person who was a referee later told me that he had decided that this just couldn't be true. And even despite the evidence and so on, but I think eventually he was convinced, and that's how I got to publish the paper anyway. So we showed by all kinds of characterizations that this was what was happening, and that you could get cyclic AMP could activate these particular genes. I did a little bit of work on myogenesis, and in fact had an NIH grant on looking at muscle development and myosin and creatine kinase during development. I went to meetings in both fields and so on. I remember Bernardo Nadal-Ginard being a very prominent figure at that time in research on myogenesis. But eventually, once I moved to Johns Hopkins, to Hunter College, I stopped doing that. So the activation of receptors by the activation of gene expression by cell surface receptors was another, I thought, was a very important finding. And it wasn't because cyclic AMP was entering the cell and behaving as a second messenger or anything, but it was because it had a specific receptor that was also the receptor that Deviotes had been studying to mediate aggregation and chemotaxis. Again, it struck me how exciting science is, and how much could be elucidated by molecular biology. And few fields provided the kinds of discoveries that we enjoyed, but on the other hand, many people would discover these things if you didn't do it. And I was not the first person to do anything like that, of course, in signal transduction. As I mentioned, Deviotes had been looking at chemotaxis, and the biochemistry had been worked out by other people as well. So my social activist side continued when I was living in Baltimore. I joined the Unitarian Church, which is a denomination I call rooted in doubt, where people embraced Christians, Jews, Muslims, agnostics, atheists, and many activists. And the Unitarians claimed as members, sometimes erroneously, a lot of people, like John Adams, who was a president, John Quincy Adams, another president, Tim Berners-Lee, who, does everybody know that? Who Tim Berners-Lee is, right? He developed the World Wide Web. And Susan B. Anthony, again, a suffragist and a Quaker. Sarah Barton, Herman Melville, Cummings, Pete Seeger, everybody knows Pete Seeger, and Whitney Young, Afro-American, progressive politician. So thinking about social and scientific issues while I was there, again, led me to another kind of episode in which I wrote this article in the Baltimore Sun. And this article bemoaned the fact that there were so few minorities or blacks in science, and that science was no big deal, that anybody could do this stuff. At least people from any race could do this. And that there were geniuses in all these populations. And what was holding us back was the access to education and opportunities and racism, of course. And there were statistics which showed that blacks earned few doctorates, 128 between 1900 and 1943, and that there were barely 1% of the total number of scientists, according to the NSF studies, and that the pool of graduate students who were black was actually shrinking. Those statistics are not much different for Latinos as well. And I wrote this impassioned article which was published in The Sun, which is a prominent newspaper in Baltimore and the nation. And George Will, does everybody know who George Will is? He's a syndicated columnist, very conservative. He writes I'm sure he has a weekly article on the upper pages of the Boston Globe, maybe even, I don't know. But a lot of papers all throughout the nation. You've heard of Maureen Dowd? Everybody heard of Maureen Dowd? Yeah, so I mean, it's like that. She writes, and her opinions are sort of published in the liberal, mainly in the liberal press, and perhaps in the Wall Street Journal too. George Will was a conservative, and he wrote back and said, "Great article." I felt kind of uneasy being complimented by him. But, and he said, "Why don't you send this article to Reagan, who was the president at that time, and see what they say." So I sent it on to the, channels. And then I got back a letter from them saying, "It's professors like you who are causing all these problems and salaries that people are demanding, raises and so on." It was totally irrelevant what they, and off base. But anyway, that article again was something that not just the impact of the people who might read it, but it really, these kinds of things transform the writer. And with my penchant for becoming involved in these kinds of things, it further stimulated that. And other people in the nation who knew about my interests and so on contacted me. So that's how I became much more involved in this educational aspect of science. And peers reacted in a kind of a mixed way at Johns Hopkins. Some of them thought it was very good and good thing to do, and others saw this as being too focused on wasting time on educational issues. And as Richard Tapia, who is an American mathematician and Hispanic, says, they made it clear that they didn't want me to be doing all these other things. It's a choice. I mean, I think that people have to make these kinds of decisions themselves. I got from my work and so on, I was recruited to advisory groups, which we called study sections at the time, and at the NIH. And at first I worked as an ad hoc reviewer, because that's how you learn the ropes and so on. And I would recommend this to all of you if you get the opportunity to do that, to be on these study sections, because these are the people who determine the fate of your grant application that you've spent months and months writing. And some of them get triaged and thrown into boxes immediately, and others get reviewed. And you need to know the process about how that works. Later I did this for a long stint, which is about 4 or 5 years, after I did the ad hoc reviewing. And few proposals would get funded. And it was just amazing. And that's how it is now. It's probably even worse. So you'd write your proposal and you try to make it as tight as possible, and you'd consult other people and collaborate and so on. And I was on this committee, and of course, being on this committee, I was the only black on the committee, on my study section. And I felt at times that, and other people indicated this to me as well, that they assumed that I was there on a racial quota, and that this, I would have nothing to contribute really. And so you would have to convince people by your arguments and your knowledge and so on. And although I felt that I knew much more than most of the people in the study section in that topic, but I would have to convince them by, through the arguments and how you'd say, well, this proposal was good and it has these particular aspects of it, whereas other proposals might be lacking and so on. And it's a very competitive environment because there's so much at stake, of course. And so, again, that induces what's called stereotype threat in the person, because you have to compensate, and it puts more pressure on you and so on. So that's a price that minorities pay for being on study sections and for being in those positions. But I would argue that that's really a small price to pay, because even if you did come because they wanted to have more minorities in the population, it's important to be there in these kinds of decision-making processes, so that you can have a role in what priorities are given to science. Should we provide more funding for AIDS, or should we provide for something that's more exotic? And all these kinds of dilemmas that face people who are in those positions. They have to decide on the future of where science is going to go. And if you're not at the table discussing these kinds of issues, then you're sort of out of the game. And for women, minorities, Latinos, and it's important for everybody to be there. And even if you feel stigmatized at times. So, and of course, the powerful institutions would have an edge, because people knew more about them, and it was easier to fund them than the smaller institutions. They had better track records and so on. So, but that's life. And the other part of what of something that I had said earlier is that you need to have communities and peer groups and people that you can rely on and communicate with, who can help you to sort of realize whether you're being paranoid about something, or whether there's good reason for you to be concerned about a particular issue, to advise you on things, and to have these kinds of peer relationship connections. And so it's very important to make these kinds of networks. And at Hopkins, I couldn't do that, except with people in the medical school, because there were more minorities there. Levi Watkins was a medical doctor and he did a lot of recruitment in the medical school. And so I went to some of their meetings and felt a kind of community and those kinds of things that are so important in your development. You don't want to be in a place where you're so lonely and there's nobody to consult about, well, this paper I'm writing, is it going to be, where should I send it, and those kinds of things. Of course, there are many, many, many other peers who are not minorities who are going to be helping you. But some of them don't realize the kinds of pressures you may be under. And also, there's always a kind of a comfort level that might be established more quickly with people of color. So that's the study section part of it. I'm going to leave out my parts about, I'll tell you a little bit about an experience I had with my son in Germany. We went to a conference. He traveled with me to many conferences, and we would sit in the back while the talks were going on, especially on a rainy day when he couldn't get outside. We would play chess. We played a lot of chess. And of course, with chess, you don't have to talk. So I'd make my move, and then I'd look at what's going on, and then he would make a few moves. He won frequently because I got distracted by the science. And when it was sunny, he would go outside, and in this particular place in Ursee, which is in Bavaria and south of Germany, we went to this conference, a genetics conference with Dictyostelium and so on. He would go outside with his soccer ball, and then all the kids from the village would follow him, and they'd start playing. He didn't speak English, they didn't speak, he didn't speak German, they didn't speak English, but it didn't matter. The soccer was a common language. So he enjoyed that a lot and had fun. And then he discovered at the back of this cluster, Ursee, a sign which was written in German, and it was obviously a cemetery, and he asked me about it, and we looked at it, and then I got it translated, and it said that was a place where they had buried a lot of the people who had been euthanized, and who lived. This cluster was once a monastery, and then it became a sort of a sanitarium, and they had lived, and the Nazis had tortured people, and many of them were sent to concentration camps, and some were buried at the back there. And I was just appalled that we would be having a conference here in that place. And it's a kind of a self-righteousness in a way, because it wasn't the fault of the people who were there. But in any case, then a lot of the American scientists I talked to also got riled up about it. And then we protested, why are we having a conference here with all this history behind it? And we spoke to the people who were managing the conference, and they said, well, this is Germany, and it happens everywhere you go. And we didn't know that the conference was, that when we booked the place, we didn't know about this sordid history. You can look it up on the internet, you'll see the story of Cluster Ursee, probably on Wikipedia. But anyway, that was a kind of another kind of moment in my scientific life where, again, I felt that, it's really, all these things that while we are working in the labs and so on, there are lots of things that we don't think about. And I was glad that my son was a part of that. I took pictures of him and so on, but I could never find the pictures. There were, I thought that was a big impact. So my involvement with Garrity, the judge, with the Baltimore Sun, with the Cluster Ursee and so on, were all episodic, individual kinds of events, like you might have events today with Eric Garner or Michael Brown, people who've been killed by confrontations with police and so on. You hear about them and they go away, and then you sort of go on with your life. I thought that I wanted to do something that was more long-term and systematic, and I had been attracted by people at Hopkins, at Hunter College, who were interested in developing an environment for minorities in science. And I went there. They asked me to come and to set up 2 2 minority scientists asked me to come, plus the president of the university, to take a job there. And I'm skipping ahead of some things because we're running out of time. And so I went to Baltimore. Donna Shalala was, anybody heard of her? She was the Health Secretary of Health.
HHW
Yeah,
for
right.
Clinton, if
Yes,
I remember.
right. She was the president.
President of the University of Miami.
Yeah. She's the she was the president of Hunter College at the time. And she recruited me. We were going to set up an environment with a lot of, where we'd be making a sort of a multi-racial environment with students and faculty from all kinds of tribes. And they would be working together and harmoniously. And that was the goal. And so I took the job. I negotiated a lot of things very carefully. And I went there, and we wanted to have a long-term program which would have some impact. And about a year later, the people who recruited me, the 2 minority scientists who recruited me, left. And they went to other places. And so I was the only one there. There was a gene center. There were funds available from an NIH grant to try to do this project. And I took it over. And we wanted to diversify the faculty and to build up the infrastructure and so on at Hunter College. And so I worked on that project, and which was, I had worked on that for many years since then. Many of you might have grandmothers or mothers who've been to Hunter College in the past, and or some of you might. There were 2 Nobel laureates who had been there. Gertrude Elion, who developed AZT, that's used for blocking reverse transcriptase, or incorporating the wrong nucleotides into the genome. And Rosalind Yallow, who developed the radioimmune assay. Also, Ruth Teitelbaum Lichterman had worked on things that were called ENIACs, and that's an electronic, numerical, and integrator computer. We now call these things computers. And they were used for, the first computers were used mainly for the military to determine how projectiles should be launched. And she was a Hunter College graduate, and she and they called those people computers at the time, because they were doing the computing, writing the programs and so on. And so anyway, it has a kind of a distinguished past, and when it was a women's college. But what attracted me was that there was an undergraduate population there that was quite diverse. 50% minorities, including Latinos and all kinds of people. I liked that. And that attracted me. I had an opportunity to go to Princeton as well, and I decided to go to Hunter. I set up a board, an advisory board for my committee, for the grant I was running. Ken Herwood was a member, John Alderetti, Latino, Max Gottesman, and Norma Allwell were on that committee. And I wrote grants to the DOD and got equipment for bioimaging and confocal microscopes and all those other kinds of things. And recruited people who could do, in order that we could get ELAC accreditation for animal care and set up better infrastructure. I wrote a grant to the NIH to build a website, which is one of the things that I thought was important, where at that time there weren't many websites, by the way.
This was the early 90s then. 88, 90,
yeah,
early 90s.
And we had, and that was for getting minorities who were like needles in haystacks all over the country to at least interact with each other. And it was called the Just Garcia Hill site because it was named after 3 prominent scientists, minorities. Just, who was a developmental biologist, Fabian Garcia, a Latino agronomist, and Rosa Minoka Hill, a Native American medical doctor. So that's what we did, and we had 4,000 people who joined and interacted and so on, and that helped us in recruiting other people people to Hunter, because we had this network, basically. One of the main elements of success, in my opinion, is to have networks. And so we got grants from the NIH, renovated labs, and Sidney McNary, who was running the RCMI program, there helped a lot. And we recruited both minority and majority faculty. Mary Philbin, for instance, who was at Hopkins at the time, came to Hunter. She just died recently. A neurobiologist who's worked on myelin and demyelination. You may have heard of her. And she was one of the people who, an Irish woman. Jesus Angulo, who is a member of the NIH council, one of the councils there, was recruited in that way. And he's now taken over my position there as the head of this gene center that we formed. We started a symposium series, and after the first symposium I took over, and we've had 25 of them since. International symposia that invite lots of prominent scientists, Nobel Prize winners. We invited David Baltimore, Bob Horvitz, and Phil Sharp, all participated in this from MIT. But of course there were others from different places. These conferences are now a significant part of the scientific life in New York annually, and you're invited to come. We installed high-speed networks, local area networks, and also we developed the first, we arranged to have fiber, which is abundant in the ground in Manhattan, unlit, lighted up so that we could have access to internet too. And we used it for video conferencing, Carlos Leoran and I. And we used that to do more outreach and to communicate with scientists by interactive video conferencing strategies that we developed. So we renewed the grant. The last time I renewed the grant was about almost 3 years ago. And I have since stepped down from that position. The last award was for 16 million. But it also aggregated many other kinds of grants that we could get as a result of that kind of growth. And we focused on improving the infrastructure and recruiting faculty through these many networks. We expanded the membership of the people who were qualified to be in it, from just biology and chemistry to psychologists, some psychologists, and some even biophysicists. And so we had all these different tribes, as I call them, who were participating in this collaborative environment that would lead to better science. And when I stepped down a year ago, I had, from one minority scientist among 30, we now have 15 among 50. So we've increased both the minority and the non-minority faculty who are involved in this enterprise, and which has a great impact in terms of productivity, grants received, conferences, published papers, and collaborations, and all the criteria that you use for success in an institution. And of course, grant funding had risen manyfold, and so had R01 grants, which are highly coveted. I didn't do much to do the student recruitment. And in fact, Mandana would be disappointed to hear that, because I know that she comes to Hunter and recruits students and so on. Some of them have come here on summer programs. I know from last year, some students told me that they'd been to MIT and met Lambda and so on, and they were very excited about that. But people like Derek Brazil and Vanya Quinones, whom I had recruited in this program, are the ones who you probably dealt with. Vicky Lewin as well. But I didn't recruit her. So there's some progress, and we made some impact, I think. And I think I have a picture of that. Let's see if I can find that. Oh, sorry. Well, I'll show you that in a while. Let me just get back to this Africa thing. Using our video conferencing strategies that we had developed, we could use from some of the classes that I held. Do I have a pointer? Oh, yeah. In my own bioinformatics classes, we sometimes do this kind of format in which this is all people in different locations. This guy is in Nevada. These people are in Africa. These are people in different parts of Brooklyn, and we're talking about some brachy or some problem. But they all have headphones, and we use software which we email to them, and they can connect. It's not Skype, because you can't do some of this stuff with Skype. And we would have these get input from different places, which is important again, because you don't have all the expertise in one place, and you can rely on other people who are willing to collaborate and to have an input and so on. And that's how we started doing that. But then we went, the H3 Africa project is one that's funded by the National Institutes of Health here, and also the Wellcome Trust in England. And there, the goal is to try to study heredity, health, and humanity in Africa, that Africans would be involved in finding out about their own origin and so on, and their genomes, which is really important. And so this is a huge genomic project that's funded to South Africa, Addis Ababa, Nairobi, Abuja, which is in Nigeria, and other places. There are about 10 sites and nodes, and these are developing to teach bioinformatics. And we've helped to set up an environment where they can communicate and do this kind of video conferencing, in spite of some of the infrastructure problems that they may have. And so there's, I think I have another slide here, yeah, which shows here's Carlos, my colleague, who was at that time in Nairobi helping them. Sorry, am I getting? Carlos here. And then there are experts from Europe who came down to Nairobi, I believe, and were giving workshops on bioinformatics, and we could relay those interactive sessions to Abuja in Nigeria and Ibadan and other places, so that they could take part in the lessons and in the conversations, and you could see each other and so on, and multi-point video conferencing. And so that's an example of a session. We've also used this with the Clinical and Translational Research Center, which we formed, well, which we were involved in, that involves, as I mentioned earlier, Weill Cornell Medical College, Memorial Sloan Kettering Cancer Center, and Hospital for Special Surgery. They were awarded a huge grant to promote translational research. And we were a part of that grant. And so that allows us to integrate our research with what's going on at those places. A very valuable relationship. And that's important because now we have these kinds of very tight relationships and collaborations with people in Sloan Kettering and in Cornell and so on. And so there's a lot of exchange because we're providing basic research, and there are people with whom we can collaborate. Physicists there have worked with physicists at Hunter, have worked with ophthalmologists to find new ways of doing imaging of the eye to look for tumors and so on. And they developed methods that are 10-fold more sensitive than pulse echo, using lasers to generate the sound for imaging. And we have nanotechnology people in chemistry department who have ways of finding cells, detecting cancer cells without biomarkers, based on their elasticity and their ability to expand in isotonic medium, which could be sensed by looking for changes in impedance. All of these strategies require contributions from Hunter scientists and collaborations with people in the medical school and in the medical schools around there. And so translational research is a very important area. Even though when we started, I got a lot of pushback from people who were saying, oh, Zahuni, who was the head of the National Institutes of Health at the time, he's in charge of translational research, and when he goes, so will translational research. And they objected because many of them had individual collaborations with people at Rockefeller or some other place, and didn't see the value of having a system in which people could collaborate within it and still have get access to patients and samples and so on, and IRBs, permissions, that you wouldn't otherwise get if you were just collaborating with one person. So we set up this kind of arrangement. And it's been, of course, done in many other colleges now. It's a good model, I think, for, MIT probably has some translational research projects, and it's a good model for integrating the research from one institution to another. And in this case, Hunter College benefited from that. And it's benefited to the extent that now our president has bought a floor on one of the new buildings in the in Weill Cornell Medical College, into which some of our people who are working on prostate cancer and so on will go. And then there's a new building that's being constructed on FDR Highway in that same area, where Memorial Sloan Kettering will have one tower, and Hunter will have a tower in which scientists will be there, and nurses, the School of Nursing. And so there's a lot more collaboration, interaction, and that's an activity that she can justly claim credit for pushing on. So those are the kinds of interactions and events that have arisen from the kinds of relationships that I described. We do outreach by, we're developing a strategy of doing preventative medicine by getting doctors who come into Hunter and sit in the studio that we've built, and they can do video conferencing to people in many different sites at the same time. And they talk about diabetes to people and in different communities, and get feedback from them, and then we try to track whether there's any, whether they have, it has had an impact on their behaviors, and whether in fact those people who have been exposed to this kind of information and interaction have transmitted information to their friends and family. We're testing hypotheses about models like that have been developed by Christakis and Fowler from Harvard about how obesity, for instance, can be transmitted, they claim, by like a disease or like a viral, from people to other people and so on, with whom they contact, and how connection works and how influence can be transmitted. And you can do that with looking at prevention of diabetes and so on. So those kinds of strategies are still being employed now. So it's been quite a long, exciting journey in science. These are some of the people, the motley crew, as I call them, of scientists who we've recruited. These are members of the Gene Center. Pictures are maybe a year or two old now. But it's a far different group of scientists than when I came there. And that's one of the things that I am proud of. And I think that could be a model for science in other places and in this country. Thank you.
And thank you so much. I think you can see why, on one hand, it was so much fun and excitement having Robert in the lab, and how much important influence he's had on science in the United States and organization of science in the United States. You'll take a few questions from the group?
Sure.
Anyone? Yes.
So I'm coming close to Harvey's lab. Can you share us some of your experience? What was the Harvey's lab looks like 40 years ago?
Oh,
No.
sure.
No.
Well, I don't know what it looks like now, but you know, it was a small, well, by contemporary standards, it was a big lab, and I'm sure it's much bigger now. And David and Harvey shared the same domain, sort of, and space. They had their offices there, David Baltimore, that is, and we interacted a lot with people. There was always somebody in the lab. people were working day and night, and it was very inspiring. And people tease me because I think the bench that I was working on was the one in which reverse transcriptase was developed. And this is a hard act to follow, right? And it was discovered there. That's where so David Baltimore had worked there and so on. They all said,
I just have to add, before you came, the first cDNA library ever was made by Inder Verma using Dictyostelium DNA.
yeah.
Just to show you where we were working.
Yeah.
But keep going.
And so it was a very exciting time. I distinctly remember coming back to the lab many times at night because I'd put my samples on an ice bucket and so on, and I had this recurring kind of event where I would think, did I put the samples back onto the into the refrigerator? I wake up in the middle of the night. It's like my childhood nightmare with the shadows on the window. And then I would get up, jump into the car, and drive off. It wasn't very far, but of course, at the middle of the night, I wanted to get back. And I'd come in, and there would be people in the lab still. And then I'd see that I had, in fact, put the sample away. And the bucket was just sitting there with water in it. And that was, I imagined it may have happened to some of you as well. But that was, or perhaps now you have these gels that last a long time, and they don't melt away, and your tubes don't go floating away in them. But there were a lot of bright people, as there are now. I mean, that's one of the great things about being at MIT. You just, there are more geniuses per square inch than almost anywhere else on the planet, right? And so you can exchange ideas and interact with people and really benefit from those kinds of conversation. It's also a very competitive environment. And that's part of doing science. I enjoyed it. I really, at times, of course, it was stressful. And then you have to get your papers out and those kinds of things, which are really tough at times. And your experiments have to work, or you need to switch topics and so on. But those are always things that I think you experience even now. Harvey was very supportive. I'm not just saying that because he's here now.
Let me ask you a question I always wanted to ask when you were leaving the lab, but never did. You interviewed at a lot of places.
Yeah.
And you interviewed at a time when affirmative action was really just beginning in the 70s. Describe some of your experiences, because I remember you coming back and telling me you weren't always treated as a scientist.
I think that's sort of par for the course for but the place I went to places like Caltech and gave seminars, and the experience was quite good. I met Davidson and talked with him, and I thought it was very candid kind of discussion and so on. There were other places that I won't mention or name where I thought, why am I here? These people are looking just to find, they want to have, say that they interviewed some minorities or something like that, and that kind of thing. And they weren't sort of interested in the science as much as I was. And my temperament wasn't one that sort of tolerated any tokenism and that kind of thing, which is why I ended up doing all the things that I did with respect to minorities in science. And it's good to see the quality and the number of people who have come in now into this arena and how diverse it is. So there were good incidents and some that were challenging. But I felt very privileged because I had already been insulated from by being educated the way I was, and it wasn't like, and I feel for African American and Latino people who are coming up and have to put up with this nonsense. I think it's diminishing a lot. There's a lot more momentum to try to recruit people and to train them and so on. But when you read the newspapers now, and you look at the kinds of things that are going on outside of the lab, in the streets, people walking down and getting arrested for, in a Walmart for having a a BB gun which he picked up from the store itself and shot. Those kinds of things are just unbelievable, unacceptable. And it's America, that's where we live, and that's our country. I feel very dedicated to it, and so it's a good and a bad.
One of the things you said which jumped out at me particularly was how you said that you wanted to do something special in science, and not just something that people could and would do immediately. I mean, I'm incoming first-year student, so I think I'm still too green to know what you're talking about firsthand, but it reminded me of something I read about Richard Feynman, how he'd always wanted to be this great theory builder, and he didn't really think much of the work that won him the Nobel Prize. He treated it as sort of like a mathematical trick, sort of exercise in virtuosity, I guess. It wasn't really a great piece of new physics. And I remember reading that his sort of regret at the end of his life is that he never became known as one of the people, the great physicists who discovered truly new physics. And so what this says to me is that even people who we regard as the greatest of scientists, among the greatest of all time, are, I guess, driven by that sort of ego or desire to be special. So my question is, what's the role for that sort of feeling in modern science? Is this something we should be wary of, concerned about, or what's your opinion on that?
Tough question. I don't know. I think I believe that you're right. That a lot of what drives you is your ego and your desire to do something unique and different, and that that's exciting to you in a particular field. I don't consider myself in the same class as Richard Feynman or anything, but I think that that's a very valuable kind of motivating element. And it's not. It can, of course, become a sort of a destructive force. But for most people, I think it's normal to have those kinds of desires and to try to seek to do something that no one else has done. And in a way, it's a little bit frustrating, but I think it's very important that your work becomes discovered or right after, or someone else does the same thing. That ratifies or confirms that it's really sound science. And I don't consider myself as being, an outstanding stellar scientist or anything, but even as I went along, I felt that was a very important part of the process. And I'm sure you will too, as you go along, because I think that's what drives you. For some people, it's more, excuse me, especially among scientists. I mean, for some people, it's money. You go into business and you set up something and you feel great about that. But for other people, it's just that kind of curiosity and playing with ideas, and that's what we're trapped in. That's what we feel. That's what we do. Yes.
I can see my advisor's pictures on in the slides.
Who's that?
Hiroshi Matsui.
Oh, Matsui, yes.
Yeah.
He's a great guy.
So I know you for a long time.
Yeah.
I have a question. I'm moving to Texas Tech as faculty. So I wonder how do we get review, proposal reviewer, or how to get this kind of advisor board? How do we get connect proposal reviewer? How do we connect program managers? What do we do?
Do you mean program? How do you get connections with people who are potential reviewers or might help you in that?
Yeah.
I mean, I think that now there's a lot more networks like LinkedIn and so on. I would advise all of you to be a member or to join up in those kinds of things. There's Mendeley. I don't know if you've ever heard of that, where you can share a lot of journals and find people who are interested in your work in different parts of the world because they have all these interactive groups and so on. Those kinds of networks are, of course, very valuable and important. Matsui is a great colleague. I spoke about his work. He's the one who had developed these methods of nanotechnology in which you can find cells that are cancerous and detect them in very small concentrations because they tend to have more elastic properties. And you can measure in hypertonic medium the changes in impedance that some cells lyse and others don't, and he's got these fantastic methods of doing that. He's looking at now for prostate cancer cells in urine, things like that. These are all very important developments, and in collaboration with people in the Weill Cornell Medical School. I'll tell him I saw you.
Robert, thank you so much.
Thanks.
And inspiring. I think you've inspired a large number of people, so thank you.